So welcome to this basic science plenary session. Again, my name is Eric Cohen from University of Montreal, and I'm co-chairing the basic science track of this meeting with Dr. Brenner. So today it is my great pleasure to introduce our basic science keynote speaker, Dr. Tom Hope of Northwestern University in Chicago. So Tom received his PhD in immunology from UC Berkeley. He started working on HIV during his postdoctoral training at UCSF, where he worked on HIV-Rev. He then moved on and did very groundbreaking work on the retroviral nuclear RNA nuclear export. During the last decade, his lab pioneered the use of imaging technology and live microscopy to study various aspects of HIV biology. His studies provided new insight into how HIV, for example, use microtubules to move inside infected cells. His laboratory was also instrumental in defining the mechanism of enhancement of infectivity in dendritic cells through virological synapse. And using, again, live imaging, he also revealed the dynamic nature of the interaction between HIV and TREM5 alpha during restriction. So more recently, his lab has begun to look at the interaction of HIV with intact epithelial barriers, which line surfaces exposed during sexual transmission. And this will be indeed the subject of his talk today. So it is a great pleasure to have you here. Tom, we are delighted to have you in Montreal and to this meeting, and I invite you to the podium. Thank you very much.
Bonjour. It's a real pleasure to be here, and I begin with my conflict disclosure slide. And to liven it up, I included some jellyfish at the museum, at the aquarium in Chicago. So to jump right in, what I'd like to talk to you about today is the work we've been doing to try to understand how sexual transmission happens. There is a great effort here and around the world to try to develop vaccines, microbicides, and other sort of interventions to stop transmission. And when you get right down to the details of it, it really isn't understood very well at all. So what I'm going to focus on today is male to female transmission. And so to initiate this discussion, we need to appreciate there are two distinct types of epithelial barriers that the virus faces during transmission. The first is the squamous epithelium of the vagina and ectocervix. This is a robust barrier, very much like our skin. And then the second barrier is a simple columnar barrier, which is present in the endocervix. So typically for a discussion of sexual transmission to women, a slide like this is shown. This is from Shattuck and Moore review in 2003. So on one hand, you have the columnar epithelium. This is a very thin barrier, and these cells are alive. And so this, because the barrier is thin, because the target cells, HIV can only infect certain cells of the immune system. Those target cells are close by. These cells are alive. This is considered by most to be the region of vulnerability in terms of the virus crossing the barrier to reach target cells. And these cells, because they are alive, can undergo processes such as transcytosis. Some suggest they might even be able to be infected. You could potentially also have infected cells or cells moving back and forth through this barrier. The other barrier is the stratified squamous epithelium, which I think is going to be on your right. That's a thicker barrier, and it's generally considered to be impenetrable. The virus can't cross that barrier, which is why very often a figure like this will show a break in the barrier, physical abrasion. The idea being then that the virus can pass through the barrier to reach either Langerhans cells, which are resident within that epithelial barrier, or underlying target cells, T cells, dendritic cells, macrophages. Before I'm done today, I will hopefully convince you that virus can at least penetrate partially into this stratified squamous epithelium, and that virus that penetrates may play a role in the transmission event. So as Eric mentioned, my lab for a number of years has been doing imaging of HIV, and the way that we began this was by taking the green fluorescent protein derived from jellyfish and essentially putting it into the virus through a protein called VPR. The details really aren't critical here, but essentially we take cells, we put in the DNA for the virus, we put in the DNA for this fused protein, and as the virus is assembled, it takes up the GFP VPR, so we end up with labeled virions. We can then put those virions into cells and watch them move around. In this case, we wanted to see if we could use this system to study how the virus interacts with barriers. So we put virus onto the tissue, we cut the tissue into thin sections, we looked under the microscope, and as you can see with the white arrows, we could see little green dots in the tissue, and we were thinking, okay, this is going to be great. Then we did our control experiments, and where we didn't put any virus on, and we still saw little green dots in the tissue. So this became a major obstacle because we couldn't tell which little green dot was a virus and which one wasn't. And we worked for a while to solve this problem, and luckily we're able to develop a very nice solution. And that solution uses a derivative of the green fluorescent called photoactivatable GFP. It's essentially non-fluorescent. It's almost as if you have a switch on the protein, you can turn it off. And then if you hit it with another color light, a nice purple color around 400 to 430 nanometer light, you then turn it on. And so that's illustrated here at the bottom of this slide with some viral particles that are stuck to a cover slip. So before photoactivation, you can see hardly any viral particles. Then after we photoactivate, and all these pictures are taken in black and white, so we pseudocolor them. So before photoactivation, we show green. After photoactivation is red. You can see now a robust signal. Then in blue, we stain for viral capsid protein, and you can see that the red spots overlap with the viral proteins, showing we can see viral particles. So now using this system, we're going to do a series of explant studies, and so we take tissue as quickly as we can obtain it from hysterectomies that take place at Northwestern University with all the appropriate IRB approvals. We cut the tissue into small, about ½ centimeter cubes. We immerse the tissue in virus for 4 hours. We remove the tissue, put it into a cryomold, section it, and then look under the microscope. These experiments are done generally under these conditions where we use replication competent virus that is labeled with the PA-GFP. The amounts here are sort of standard in the laboratory, 300 to 500 nanograms per mil of P24. But as I'll discuss later, this is especially compared to a sexual transmission event, is a lot of virus. And then we will compare what's going on in terms of the virus interacting with tissue in matched ectocervix and endocervical explants. So here's an example of that sort of analysis, and just kind of quickly what we do is we take the section, you scan through it in the green channel to define the background, you photoactivate, and then you do the second scan exactly the same way. And so what you look for is a signal that appears on the second scan that's in red that isn't present in the first. And here you can see in this section of endocervix 2 red dots. Those are the viral particles. The yellow that we usually think of for this sort of analysis, co-localization, the background co-localizes. So here is evidence of 2 viral particles penetrating the endocervical columnar epithelium. And in this case now we're going to switch the green color to cytokeratin 7. It just shows, that these are columnar epithelial cells and gives you an idea of the boundaries of the cell. And you can see that these viral particles are located within the cell, consistent with the idea that they can be endocytosed and or transcytosed. When we did these sort of experiments, we often saw, and you can see on the left-hand side, the virus is sort of hovering above the tissue. We were a little bit confused by that. But it turns out that this columnar epithelium is coated with a layer of mucus. So to see that mucus, we can just stain with wheat germ agglutinin, which gives us this green haze of sugars present above the columnar epithelium. And we believe that mucus is probably playing an important role in the transmission event. So to test that, to demonstrate that, here's an example of an experiment where we use neuraminidase to deplete the mucus, the protective mucus layer. So in the upper two panels, you can see in panel B, we stained for mucin 5AC. So the green color is the mucus, and the virus, as seen with the little white arrows, is essentially held away from the underlying epithelial cells by this mucus layer. If we use neuraminidase, which cleaves the terminal sialic acids off of the mucins and causes the mucus structure to fall apart, you can see then in the lower panel that the virus is now able to reach the underlying tissue. We can quantify this, and so you can see that either with BAL, an R5 tropic virus, or BRU-D, which is really a virus without any envelope, we see an increase in the amount of virus that's able to reach the tissue when we deplete the mucus. So as one would suspect, the mucus is able to play a protective role in keeping virus away from this potentially sensitive single layer of columnar epithelial cells. So how about the squamous epithelium? So here is before and after photoactivation of an example. The viral particles that are penetrating the tissue here are seen within the white circles, and we can see that the virus is able to enter the squamous epithelium. If we again stain for sugars just to get a sense of the tissue structure with wheat germ agglutinin, we can see that the viral particles are not located in between inside of the cells as we see for the living columnar epithelial cells, but instead is typically found in between the cells. In this case, we quantified it, and 90% of the time, the virus penetrating the squamous epithelium is in between the cells, and I think this is telling us that the virus is sort of moving in by diffusing in between the cells rather than interacting with the cells in some other way, because these cells at the surface of the squamous epithelium are essentially part of a layer of dead cells that provides a barrier. So one of the things we were very worried about, this was an unexpected result, was that from the moment of surgery, these tissues are falling apart. They're degrading probably to some extent. So what we wanted to do was confirmatory studies in a rhesus macaque model, where we apply the viral vaginally while the animal's alive, use the same supernatants with the GFP labeled viruses, wait 4 hours, sacrifice the animal, and then look and see if we see the virus entering the tissue. In fact, we do see the virus entering the tissue, as you can see here within the white circles. Again, we stain with wheat germ agglutinin, and again, we see that the virus is in between the cells. In this case, when we quantify it, it's about 50% of the time the virus is in between the cells. The rest of the time, it's kind of indeterminate. At the surface of the macaque, unlike a woman, the cells are very compacted. It's almost keratinized, and so it's very difficult to tell where one cell ends and the other one begins. So there are a lot of indeterminate virions in terms of whether they're between cells or not within the rhesus macaque model. So we now can take a very large data set where we have looked in comparing the tissue from endo and ecto in women from, I think it's getting close to 60 donors now. And we can look at the distribution of the thousands of viral particles that we've quantified. And what we do is we take each particle we identify, and then we measure the closest distance to the epithelial barrier. And so on the y-axis is the number of viral particles counted. On the x-axis is the depth of penetration. And so what you can see is that most of the viruses, the darker color, the purple, is ectocervix or squamous. The lighter color is endo. And what you can see is that most of the virions are penetrating on the order of up to about 10 microns, and that's the thickness of that columnar epithelial layer. But some virions are penetrating quite deeply into the system, 40, 50 microns. And in fact, when we do this analysis, the field of view is 60 microns. So in this sort of a study, we can't detect the virion penetrating deeper than 60 microns because it would be outside of our field of view. We have to include the barrier. So I can say that in some cases we see the virus penetrating even more deeply. And I don't have time to show you the data from the individual donors, but we see virions penetrating deeply into the tissue in explants from most donors. So this isn't one or two donors. Somehow the virus is penetrating deeply, but very often the virus penetrates deeply into the tissue. So an important point that I want to make here is the barrier works most of the time. When we take a field of view, one nice thing about this system with the photoactivatable virus is you do truly a random survey. We can't tell if there's a viral particle there until you initiate this before photoactivation, after photoactivation. So we really get a nice random survey. And what this slide shows is that about 87% of the time when we look, there are no viral particles. But then the minority of the time, 13% of the time, we see one or more particles entering the tissue. And I think what that's telling us is that generally the barrier is working, but once in a while there's a problem with the barrier, and that's where the virus can penetrate this protective layer of epithelial cells. So to try to get insights into why the virus is able to penetrate in some places, we began to look at the proteins that help to form the epithelial barriers, adherens junctions, tight junctions, desmosomes. And here's just an example of that from the rhesus macaque model. In green, you can see ecadherin, which are adherens junctions, and you can see that there is robust expression of adherens junctions within that spinosum layer of the epithelium where the cells are dividing and differentiating. This also illustrates in the macaque the very thick layer at the surface, where essentially made up of dead cells. In that layer, there are no adherens junctions, as you can see from the lack of signal, or very few adherens junctions. There, the predominant structure holding the tissue together are desmosomes. So to get a sense of how these tight junctions and adherens junctions and desmosomes were influencing the barrier function, we used a fluid phase marker. So in this case, just fluorescently labeled BSA. Fluorescently labeled BSA is in the water or the media, and if water can penetrate the tissue, the fluorescent signal will go there and show us how waters can enter. And so here we have the BSA is in green, and so you can see water is able to penetrate the most superficial layers of this cervical, ectocervical explant. In red is the adherens junctions, and what you can see is a reciprocal relationship. So in that one spot where the adherens junctions come right to the surface, there's no water entering the tissue. So the adherens junctions, tight junctions, are excluding water, but the most superficial layers where those structures are not present do not exclude water from entering the tissue. We see the same thing in a macaque. Now this is a much larger area of view here. This is probably about 1 millimeter of tissue being looked at. And what we can see is in some places the barrier function is working really well. The BSA does not enter the tissue, but in other places you can see the red signal entering the tissue quite deeply. And so the barrier is not working in all cases. So this heterogeneity in barrier function is really similar to the heterogeneity in virus penetration we saw earlier. So we did an experiment where we asked the question about virus localization in a different way. In this case, what we did is we said, okay, if BSA penetrates the tissue less than 5 microns, where the barrier is good, how many viruses do we count there, versus in places where we see the virus penetrating deeply into the tissue? I'm sorry, the BSA is going more than 5 microns into the tissue where the barrier is not functioning as well. What happens there? And to just sort of go right to the answer to save time, in the box there where it's penetrating virions per Z-scan, we see a 10-fold increase in the ability of virus to enter the tissue where the water is able to enter the tissue. So I think this has a number of really important implications for understanding sexual transmission. The first is, and I can't show you all the data, is that we can take nanobeads or virus R5, virus X4, virus without any envelope, and it just goes with the water. As the water enters the tissue, the virus goes along. So it seems like the penetration of the virus is simple diffusion, and it's even a little more than that. If there's also an absorption aspect to this tissue. The environment is moist but not saturated with water. And so from experiments, for instance, where we take the tissue and put it into media overnight and then add virus to it, we don't see much virus entering the tissue, if any entering at all. Whereas if we add the virus with media at first, we see this absorption of water into the tissue, drawn in, sort of filling in the gaps in between the cells. That's when we see the virus entering. So, so this appears to be the mechanism of virus penetrating the tissue. So hopefully I've convinced you the virus can enter the tissue up to 50 microns or more. And so is it possible for the virus to bump into a target cell to initiate infection? This graph shows the penetrating virus compared to either Langerhans cells or T cells. And we see that they are going to both be in the same area within the tissue potentially to be able to interact to initiate an infection. And then this is typically CD4 cells aren't abundant within the epithelium. But one of the things that can cause infiltration of T cells into epithelium is inflammation or other sort of circumstances. In this case, we're looking at tissue from, and this is actually one of my favorite images. This is a really beautiful image of a depot-treated macaque where they're treated with Depo-Provera. The epithelium is thinned, and under these circumstances, we think that the immune system within the tissue now can better sense the outside environment. They sense that outer environment because the epithelial barrier is thinner. They sense something that's different, so you get this infiltration of immune cells, this infiltration of T cells. And as you can see here, some of the T cells move up into the epithelium right to the surface, where a virus just interacting with the surface of the epithelium or penetrating slightly would bump into a target cell. Here's an example of a local site of inflammation. We find these very regularly in the ectocervix of women that are donating tissue to us. And likely this would not be associated with any sort of symptoms. It's just a small area where we have, and again in green here, CD4 positive cells infiltrating into the epithelium. So our model for transmission when the tissue is intact is the idea the virus is penetrating the epithelium, and it's penetrating as a gradient. The deeper you go into the tissue, the fewer virions are present, and this is also very focal and related to barrier function. They can penetrate to the level where they will bump into Langerhans cells, but we know that probably the number one confounder of acquisition, the number one way to increase one's odds of getting infected is inflammation. And this is true for women, men, and even mother to infant transmission. And so you get infiltration of target cells into the tissue. The virus potentially does its same thing, and you just have an increase in the odds of the virus bumping into a T cell and initiating a transmission event. So we now, I think, have a pretty large data set. We're seeing pretty consistent results, and we're starting to have an appreciation for the underlying mechanisms. And so what we've been trying to do lately is to see if we can model what we're observing in macaques and in explant cultures to what happens with during sexual transmission. And so we're going to work with numbers here, and I went into biology because I didn't like math. Math is very exact. Numbers are numbers. It's 1 or 2. It's not something else. And so but we have to do this, and so this is my best effort to try. So I'm going to start with caveats, and the first is I am not a mathematician or a modeler. I'm very interested in talking to real ones about this, so please give me your feedback. We're going to assume that the ectocervical explants, because we can't really work with vaginal tissue in humans, we can't get access to the tissue is roughly the same as what we see for the ectocervical explants, and at least for macaques where we do get the vaginal tissue, they're only about 1.5-fold difference. So a little more virus penetrates the vaginal epithelium. And then the other caveat is that the interstitial penetration we observe can actually lead to a transmission event. So then I think we have to have some assumptions here. The first is that because the virus is going in with the water, and the water is going to go in, whatever concentration of the virus is present in the liquid is going to penetrate the tissue. And this actually is borne out in some data I didn't have a chance to show you, where there's a statistically significant correlation between the virus concentration we add and its ability to penetrate the tissue. So we're also going to assume that semen is not making a big difference in our results. We have done some explant studies with viral absorption in the presence of semen, and it doesn't have much of an influence. And then also the 4-hour time point that we're basing this on sort of gives us a maximum value for penetration of the number of viruses and the depth they're traveling. All right, so virus penetration of the vaginal vault. If we take our complete data set, and we're using an average concentration of 500 nanograms per mil of virus here, this gives us 1.21 virions penetrating the tissue per each section, or each region imaged. As I said earlier, that's 60 square microns, and it's also 12 microns thick. So the surface area of there is 720 squared microns. So if we look at, if we divide those two, we have a very small number of viruses per micron. So we're going to then multiply that times 100 million to give us the number of virions per centimeter squared. So that's 168,000 virions per centimeter square. And then the average surface area of the vaginal vault, as calculated in the paper at the bottom there, is 85 squared centimeters. So about 14 million virions are penetrating if the viral concentration is 500 nanograms per mil. But that's not the concentration. So the highest reported concentration in semen is 4 million virions per mil. This was in the acute phase, and this is by far the highest number ever detected. In a chronic circumstance, we're dealing with about 5,000 virions per mil. And I think in the acute phase, it's up into 30 to 50,000 virions per mil typically. 1 picogram of P24 is 7,900 virions, assuming 3,000 gags per virion. And so the number in the lab, and this is very common in the lab, 500 nanograms of P24 is almost 4 billion virions per mil. Right? And that's a big difference from the chronic phase where there's 5,000 per mil. So if we use the largest number ever seen for virus and semen, and per exposure event, there'd be 14,000 viruses penetrating the tissue. And if there's only 5,000 virions, there'll be 18 virions penetrating the tissue. 18 virions per exposure. That's a small number. That seems like a number we can try to stop. It's just kind of a comforting number. And especially when you consider that we don't know if all those virions are infectious, we don't know many things about those individual virions. And then it also has a number, I think, of interesting implications, because everyone sort of goes, there's these numbers that are out there from epidemiology, like 1,000 coital encounters from a chronically infected person to their spouse with any STIs and all of this. Well, if there's 18 virions each time, that number, I think, actually makes a lot of sense. Also, there's a lot of interest and the knowledge that one virion tends to establish transmission in humans, and that sort of led to this idea that there's a sieve or a filter or something like that going on. And I think there is a sieve, and the sieve is there's hardly any virions. So if there's hardly any virions and one gets across, it's usually going to be one. So what about the endocervix? That area, that is part of this vaginal vault calculation of the surface area. But I'll just say a couple things. One is, as you can see in the right, it's sort of most of the surface area of the endocervix is protected by mucus, and it's sort of sequestered deeply within the tissue in these folds. and there really is a lot less surface area there. So I think that only a few virions under these conditions would be bumping into the endocervix, and we have some more work to calculate there. So, and then consistent with this idea, if we look in the rhesus macaque, I think it's your far right, if we look in a living macaque to the far right of that box, very few virions are entering the endocervix, which struck us because it was different than on the left with human explants. And if we look in what we did then was some macaque explant models and our experiments, and we saw very similar results to the human explant. So this again confirms the idea that the endocervix, this tube full of mucus, does a good job of protecting those potentially fragile tissues. I'm not saying that I think in fact we run into trouble because the scientists want it to be one answer. The scientists care, is it more in the endocervix or the ectocervix or the vaginal vault? The virus doesn't care. The virus just wants to interact with or needs to interact with a target cell to initiate infection. So I just want to, in the last few minutes here, talk just a little bit about trying to intervene with vaccines, with antibodies. And what I want to talk about is alternative functions of antibodies rather than the neutralizing antibodies. Broadly neutralizing antibodies would be great. We're starting to really understand them, and I hope that someone can figure out a way to make a vaccine that generates them. But I would like to call for a plan B where we consider other things that antibodies could do to intervene. And this is a little bit inspired by the controversial RV144 trial in Thailand, where there may have been some protection, and if there was protection, it wasn't by any mechanism that was anticipated. There were no good T-cell responses, and there were no broadly neutralizing antibodies generated. What I'd like to talk about is where are antibodies within the female reproductive tract, and I'd like to come back to mucus. This is actually cervical mucus that has been sectioned and stained like we would do tissue. And what we can see in blue is IgA, and I think if we get the mucus that's coming from the cervix very early on, because the mucus is hydrating, it's dispersing, so this is, before it gets to do that, we see these ribbons of mucus where IgG, which is coming from transudates in the vault, is primarily painted on the outside of these ribbons, and IgA is present throughout. We think the explanation for this is that the mucus is originating from goblet cells within these crypts and folds in the cervix, and as they come out, so as they're being generated, IgA is present there, and as they move out to the surface, a little bit like when you look at a glacier and you can see sort of the way the different flows of ice are coming together. Here we see the same thing. These different ribbons are originating in each one of these crypts or folds. And then as they reach the surface, they're getting painted with IgG. The IgG on the surface and not throughout suggested that IgG might be interacting specifically with the mucus, which is something which is not known in the literature. It's known that IgA interacts with mucus, but IgG does not. And again, the idea here is if we could take mucus and essentially put antibodies on it so it has specificity to bind, you can capture virus in the mucus. So that movie just played pretty quickly, but we can use a method called FRAP to look at the dynamics of proteins. And so we're going to stain the antibodies in mucus, and we're going to do a photobleaching experiment. And the way the signal recovers after the photobleach will tell us if the antibodies are stuck in the mucus or not. So you can just see those two boxes here where we bleached the antibody signal in the mucus. We're going to do a second bleach right there. And so you can pretty clearly see it's not recovering. So we can quantify that, and you can see that the initial bleach decreases fluorescence, and it doesn't really recover over this time scale, which is about a minute. We bleach again, and it still doesn't recover. We can do this on longer time scales. So this really is suggesting that IgGs, antibodies, can be associated with the mucins, and this might be a strategy for developing a vaccine. And so here, then, what we can do is take two colors of virus, red virus that could bind to antibodies, green virus that cannot. We also sometimes include nanobeads. We put those into mucus. We watch them move around. We have a computer identify each particle and measure the distance they're moving from each frame. We can then use physics to follow and quantify this movement using plots of mean square displacement over time. And the way that you do this is you just measure the distance from where each particle is at each time frame. And then you can put those on a log-log plot. They use the second time frame, so between 1 and 3, 2 and 4, etc. And you can get these plots over time. If the slope on this plot is less than 1, it's hindered diffusion. If it's 1, it's free diffusion. If it's more than 1, it's active transport. And then depending on where it is intersecting the y-axis, it tells you how fast that free diffusion could be. But so, say a particle would move much more freely, say in water compared to glycerol or oil. But it could still be free diffusion. So I'll just show a couple of examples. This is showing if we add VRCO1 to the mucus, and kind of consistent with the idea of the way antibodies work, we see an effect at a specific concentration. So what we can see here is that the pegylated beads don't really change in their motion, and the delta envelope virus doesn't change in their motion, but the wild-type virus is slowed by the presence of VRCO1 at 0.5 micrograms per mil. We can do the same. Well, PC doesn't like that one, so we'll just keep going that one. Okay, so we can use mixtures of antibodies and see a similar phenomenon. And in this case, what we did was to use IgA, which interacts with the mucus through secretory component. And so if we just add the IgA, it doesn't really do much. But if we add the IgA and let it interact with the mucus overnight to come to equilibrium, we now can see a slowing of virus movement. And then just very quickly, I think I have about 2 minutes left. I'm just going to talk about 2 other possible things that antibodies could do. In the upper right hand, an IgA or an IgM could cross-link the virus, and we've just developed an assay to be able to look at, instead of using 2 different viruses colored differently, we use the same virus colored differently, and if it gets cross-linked together, you bring red and green into that subdiffraction area, and it becomes yellow. So this just shows a larger aggregate of virus made by antibodies, where the red and the green particles are brought together. And then we can quantify this, and it's just an example on the bottom where IgA at a certain concentration can cause overlap of red and green. So it's microscopy based, but we're developing this into a high throughput assay to look at antibodies that could potentially inhibit virus transmission by cross-linking viral particles. And then the last one I'd like to point out is this is ectocervix from a woman. And we can see IgA and IgG in the superficial layers of the tissue. Here's breaking it apart to ecadherin, IgA, and together. And what you can see is that the superficial layers, that the same way the water is penetrating the tissue, the antibodies are penetrating the tissue, and they seem to be interacting in some way because they're fairly stuck there. So with Paul Johnson at the New England Primate Center and Harvard, we did an experiment with animals that were vaccinated with an attenuated virus, the sort of famous Delta Neph virus. And when we did that experiment and we measured the distance, we were able to see a statistically significant decrease in virus penetration into the endocervix and vagina when the antibodies were present, suggesting that might be another mechanism that we can explore. So here's 3 different ways that antibodies that can bind to the virus but maybe not neutralize it might be able to function as a vaccine. So I'll go ahead and stop there and acknowledge the people in my lab and the various funders and the many collaborators, and also thank you to the individuals who have donated materials for our work. And I'll go ahead and stop there. Thank you so much.
So thank you, Tom. I think that given the schedule, we're going to stop here, and I invite you to, if you have questions, to ask them to Tom. He'll be around for the rest of the meeting. So right before finishing, I just want to give Tom a small present on behalf of the meeting organizers. Yes.
Oh, thank you so much. Wonderful.
And thank him very much for this enlightening talk. Thank you.
Thank you.
So thank you for the presentation. There is a seat in the front for those who remain by the door. Please welcome. We can take 1 or 2 minutes to invite people to come, mainly on this side of the room. Yeah, good. Yeah, we need a few minutes to let the people, because it's yeah, you like a star. People look at you, but you don't see anyone.
Yeah.
Yeah, that's the star system. You have to get used.
Yeah. I have a very high myopia, so it's very...
It's eye-opening. Please, many seats remain free by the front, so don't be shy and come. Okay, so we can start. And now I'm inviting Dr. Nikita Pai from McGill to introduce the epidemiological and public health science plenary.
Thank you, Dr. Ruti. The next plenary speaker in the EpiPop Health track is the internationally recognized expert on diagnostics, Dr. Rosanna Peeling. In this world of diagnostics, Dr. Peeling does not need any introduction. But for the lesser mortals like us, we need to know what she's been doing for a long time. She's flown in from Brazil, and we are very fortunate to have her speak at CAHR today. Dr. Peeling is currently Professor and Chair of Diagnostics Research at the London School of Hygiene and Tropical Medicine. She's had a very successful career. She was the chief of the Canadian National Reference Lab for Sexually Transmitted Diseases, followed by an 8-year stint at the WHO TDR in Geneva, where she laid the foundation of evaluative research in point of care, where there was none. This seminal series was published in Nature Reviews and cited by scientists even today. She has advised the governments of the United States, various countries in Africa, Latin America, Asia, on various aspects of implementation of point-of-care diagnostics. For all her incredible success, she remains a very modest and unassuming Canadian. On behalf of the Scientific Planning Committee, my co-chair Julie Bruno, and myself, please welcome Dr. Rosanna Peeling. Thank you.
Thank you very much, Nikki. And I really like to thank the organizing committee of this conference for inviting me here. It's really great to be back in Canada amongst friends and get caught up with what everyone's been doing. Over the next 35 minutes or so, what I'd like to do is to go over with you the HIV/AIDS global targets and challenges in meeting them. I'm on the WHO expert committee on diagnostics, and we see a lot of problems in many countries still of getting people tested. There's been a lot of technological innovations in diagnostics in the last 5-10 years or so, mostly because we advocated for companies, big and small, to invest in diagnostics that are appropriate for the developing world. And so I'll be taking you through some of those. And then after we have these products of innovation, we actually need innovation in delivery mechanisms, because many countries don't have, are not as fortunate as Canada to have really good infrastructures for delivery of diagnostics. And I'd like to say that just disclosure of I do not have any conflict of interest, and when I mention companies, it does not mean that the London School endorsed them, nor the WHO. Now, the global targets have been set at the United Nations General Assembly in 2011. And it was estimated that we would need about $22 million in order to place 15 million people on ART. And the target is to, by 2015, reduce TB deaths among people living with HIV by 50%. And we like to eliminate the mother-to-child transmission of HIV. And in some regions, such as in the Americas, there's a joint elimination of mother-to-child transmission of HIV as well as syphilis. And we also like to be able to intensify HIV transmission. This is a slide that I actually took from the head of the WHO HIV department. He made a presentation to the expert group on the progress towards ART coverage in the last few years since 2003. So it seems it's very promising that we've actually increased the patients receiving HIV from 3% to 47%. But we still have a significant gap in getting another 53% of patients that are estimated to require ART on treatment. And we also realize that an estimated about 11 million HIV-infected individuals have no access to CD4 counts, so they cannot actually be eligible for treatment, and that's part of our problem. Now, this is taken from the publication from the Treatment 2.0 initiative that WHO and UNAIDS is advocating. The goal is to achieve and sustain universal access and maximize the preventive benefit of ART by doing 5 things, optimizing drug regimens, of course, but also promoting diagnostics at the point of care as much as possible and using very simplified algorithms for getting them used. We need to overall reduce costs because at this time of economic crisis, who has $24 billion they could spare? We need to adapt delivery mechanisms to make it as easy as possible in health systems that are very, very fragile or broken. We need to mobilize communities and continue to advocate for human rights. Now, when we come to the strategy, we realize that much of it depends on diagnostics for getting people tested, for monitoring what they're doing, for looking at co-infections like TB, like HPV and HCV, and also in pregnant women, getting them tested. And so if you look at the whole program, we have these questions that we ask. What is it that we could introduce into these systems? How will we introduce them? And when you have new technologies, do these countries actually have a mechanism of assessing these technologies and say, within our country, at the point of the epidemic that we're in for HIV, where should we deploy these technologies and when can we start? There are many, many priorities often in developing countries other than HIV, and like clean water so that children don't die from diarrhea, there's malaria, etc. So there are many, many competing priorities, and trying to get diagnostics into these systems is actually quite a challenge. So what we wanted to do in the short term is to establish areas of optimization for early infant diagnosis, CD4 and viral load, and also establish operational guidance on how to deliver those. In the medium and long term, we'd like to be able to ask industry if they could actively develop products that would be useful, especially in bundles or as duplex tests, so that it simplifies the way that the diagnostics can be used. But it's quite often very difficult to ask companies to start on development of a new platform or a new diagnostic when it's very costly and there are many risks. And then at the end, when they have a good product, we turn around and ask them to donate it to the developing world. And so that's a non-starter right there. So we need to find good business mechanisms that would allow us to do the stimulation. Now, in terms of the bench to bedside pathway for development of diagnostics to getting them used, you could see that along this pathway, the R&D quite often, depending on whether you have an entirely new platform or you're using existing platform, the development quite typically takes between 2 to 10 years. For example, many of you may be experienced with the Cepheid's gene expert platform, which has now become available for TB and rifampicin resistance. That platform took about 11 years with lots of money from the U.S. government because it was after the terrorist attack of 9/11 that the government realized that they need some very, very highly accurate technology to rapidly detect agents of bioterrorism. So Cepheid was one of the companies that received that funding, and now they've come up with a really good platform that could give you highly accurate TB diagnosis and rifampicin resistance in an hour and 45 minutes. And it literally can be used at the point of care because you just put the sputum in and walk away. Now, so that took about $120 million and 10 years' time, 11 years' time. So we're now at the proof of principle stage, and they've gone through a lot of field trials. But if you look at downstream, regulatory approval in most countries take between 2 to 5 years, and especially when it's an imported commodity. And then policy and uptake takes another 5 to 7 years because policymakers quite often would like to see a lot of evaluations in the field, and also they like to see systematic reviews that would tell them that this is actually a useful test. And then even after the 5 to 7 years, even when the test is on the ground, there are quite often in the developing world problems with quality, maintaining quality of test, quality of the test system, as well as supply chains. Now, so if you think that for TB, what we are facing with in the world today is 1.8 million people die each year of TB. And right now, the Gene Expert platform is available. It would still maybe take another 7 to 10 years before they could be widely used in the developing world. And so 18 million people may die before we figure out how to get them approved in the system as well as the uptake. So this is a scenario that we hope that we could start to work together and shorten or have some other means of making the innovation of delivery work for the developing world. Now, with regard to regulation of diagnostics, when I was in WHO, we did a survey of the 191 countries that was in the UN system at that time, and 85 countries responded. And out of the 85 countries, over half of them do not regulate diagnostics. And you could see within the different regions, for example, in Africa and in Asia, in the Western Pacific region, many countries don't regulate diagnostics. And even in those that do, the standards for how they are evaluated in terms of safety and effectiveness are different. So we have now this scenario that is very, very challenging for us to introduce diagnostics into any country, because regulatory regulations are very highly variable from country to country. The approval process takes a long time, and also it's not transparent. Companies find it very hard to get information for each country that they want to apply to, what they need to do in order to get approval. A lot of approval processes don't have good standards, and there's limited success in terms of harmonizing regulations so that companies wouldn't have to do so many tests, so many clinical trials. So the scenario is that tests are sold and used in much of the developing world without any evidence that they work. And these are in countries that don't regulate diagnostics, where you just pay and register and then you could sell. And companies with good tests are unwilling or unable to compete in markets that are flooded with bad tests, because low quality tests tend to be cheaper. And now in one of the companies that have the first point of care CD4 test, this company is having to go through 29 trials in Africa alone and 42 around the world. This company originally targeted their point-of-care CD4 test at $6 per test. And because of all these trials that they have to do around the world, the last quote that we got from this company from Tanzania is that they are charging the Tanzania national government $35 per test. And that makes the Tanzanian government feel very difficult. How can they roll out a test for CD4 at $35 when they have so many other priorities? And the average cost of the average health dollar per person spent in Tanzania is around between $4 to $6 a year. And so we need to do something about harmonizing regulatory approval. The other thing that may sometimes affect Canada as well is that companies then, because of all this complexity and also the costs of regulatory trials, just don't bother to target smaller markets. And for some diseases, Canada is a smaller market. So what we found is that in many countries in Latin America, I just found this out at our Brazil meeting, they have to pay as much as 4 to 6 times the price for some commodities, because they have to go through many layers of middlemen in order to access the diagnostic from these companies. And also, so a test that would be that would cost the Canadians between $4 to $6, they're having to pay about $30 because of going through the middleman, not because of the cost of goods. And so Grand Challenges Canada, together with the Gates Foundation, has commissioned a number of us to actually look at this challenge. Now, I'd like to go through with you some of the new technologies that are coming along. And so first amongst them, there's been a lot of discussion at this meeting about oral tests versus blood-based tests. And we all know OraSure have been working for years on oral tests for HIV as well as for HCV. And Nikki has actually done a systematic review of the many publications on oral fluid versus blood specimens for HIV testing. The bottom line is, although the pool sensitivity and specificity are pretty equivalent with the two types of specimens, in a low prevalence setting, the positive predictive value is of concern to those of us in those settings where you're looking at very few, probably very few real positives amongst maybe a lot of false positives. Now, we also are concerned about the fact that you could actually, off the internet, you actually have advertisement like this about home testing. And quite often these advertisements include all kinds of approvals from the FDA, from the EU, et cetera, et cetera. But because what you do with a lot of these internet testing companies is that you just pay and you send your specimen in, and then you get an answer back. you really don't know what test they do, how they do it. It could be, somebody's kitchen where they just do this on the kitchen table. You have no idea. But of course, because of the nature of STDs and HIV, confidentiality, stigma, etc., these are actually well subscribed. And so we're concerned because we've bought some of these tests and they actually don't work. Now, what of CD4 technology? I think in Canada you're very familiar with the big equipment that has been around for years now. The BD fax caliber is a really good test. But it costs, about $75,000 to buy the machine. But because of batching, it's between $3 to $7. And then CAHR also have an equivalent machine. Now, as there's a demand for smaller machines, Partech has got a machine that is only $27 and also about $3 a test. And it still gives you a turnaround of 60 minutes and can do about between 200 and 400 specimens a day. So this is in a high throughput setting. Now, for the developing world, companies have responded and started to downsize their machines with lower throughput so that we could put the machines into lower levels of the healthcare system. And so these few machines are can do less than 50 samples a day. And typically the time has now been reduced to about 35 to 45 minutes for each test, and the costs remain at $3 to $5 because of batching. Now, when we then ask companies to actually have portable cytometers, they've responded with smaller machines that could turn around the results in 8 minutes, and is less expensive. And the one that I was telling you about that's commercially available now, that is really point of care, is the one that's made by Alair. It's called PEMER. And it's now the machine is only about this big, and it's $6,000. And the original target was $6 a test, but now it's any range between $6 and $35. The good thing about this machine is that the cartridge is very small, and it includes the lancet. So you literally touch the cartridge to the fingertip of the patient, and it would extract the blood. So there are no external supplies needed, no capillaries, etc. And then you could just lay it down on the bench for 20 minutes. And then you put it into the machine to read. And because the reading just takes seconds, it's very energy efficient. So that machine can use battery power. You could actually plug it into your car battery. It could use solar, etc. And so that is now a truly, point of care device. And we wish that the company would actually make it available widely, but there's, of course, problems with getting regulatory approval, as I mentioned. Now, what's in the pipeline? UNITAID is a UN organization that gets its money from a small taxation on airplane tickets. They've actually mapped the pipeline, and there are a number of truly point-of-care devices that are coming up in this year and next year. And a lot of those point-of-care devices have been funded through the Gates Foundation CD4 initiative. And there are machines that have a reader and, as I said, also a cartridge that includes the lancet. But there are also there are also tests that don't require a machine. For example, the Cyomix test is literally like a dipstick test. And it needs a small spinner tool, but that's not like a reader at all. And it's semi-quantitative, and it requires reading by eye, and so that may be one of the limitations. They're still to be evaluated. MBio is a small company in Colorado, and they have not only an HIV CD4, they also have HCV, and it's all done in a small reader that allows you to take a picture of the test for quality control as well. And for viral load, unfortunately, we don't have point-of-care technologies available yet. And also for infant diagnosis, we're still requiring a small machine or a large machine, and there won't be any new ones that would be available until late 2013-14. Now, one thing that I want to say about this new platform that's freely available is that the company has now a whole lot of menus of tests that can run on this machine, including they already have MRSA available, they will soon have viral load available, and they're looking at other C. difficile and other applications. And they could sell machines that are small or big, depending on your throughput. And also, what's new about this platform is that it's random access. So you don't have to have a batch of TB tests and then a batch of HIV tests. You could actually put in a TB test in 1 minute, and the next minute, if a person comes along and you need to do chlamydia and gonorrhea, you could put in that one, and then another one 2 minutes later. So each unit is a complete random access. And also, you no longer need to have a technician come and do maintenance on these machines. The company has evolved a system where they could send you a quality control cartridge, which you then put into the machine. You run the machine, and it will tell the company whether all the parts are working well. So this is a new way, a new generation of tests that in terms of point of care is very reassuring in that you no longer need to have a service contract with the company having to send out thousands of people into all different healthcare settings to do maintenance on these machines. And so right now, there's still a problem with the cost of these tests for the developing world, because right now the company is putting as a developing country price $16 per TB test, and in the developed world it's $65. So there's a big price differential. And for viral load, they haven't actually put those prices down yet. And so we're expecting that as they develop many menus of many tests along this platform, that the cost of each test will become cheaper and cheaper. So in terms of efficiencies and economies, what we like to see is HIV, TB, hepatitis B and C all multiplexed into the same test, or that if you need to do crypto or other diagnosis, that it could be bundled even if it's not the same test. And also toxicity monitoring. The opportunity is now that we could actually tell the developers what we want and what we do. So I would be very interested to hear from all of you what is it that we could bundle, we would like to see bundled, and what we would like to see in the same test. Because if you require different specimens, obviously you cannot put them into the same test. But if you require, if your testing interval for different tests are different, then it doesn't make sense to have them multiplex in the same test. So we're still trying to figure out in many countries what would be the most optimal package for bundling and multiplexing. There is now, I just came from Brazil, as Nikki said, and the Brazilian government has actually paid $4 million for these new technologies so that they could have the technology transfer and local production of this technology in Brazil for the public health system in Brazil. The idea of the dual path technology is that instead of the single lateral flow device, which actually has problems with many complex molecules competing to bind to target ligands, is that if you have a separate path for specimens and a separate path for your reagents, that you get a cleaner interaction between the antigen and the antibody, and it would increase the sensitivity of the device by between 10 to 15%. And with this also, there's the opportunity to put in multiple bands. So right now they already have a test that is for HIV, HCV, and syphilis. And there's the opportunity also with the Brazilian government having this non-exclusive license to actually be able to apply this technology to parasitic diseases and other diseases that are important in the Latin American region. There's also, last July, a lot of press about this new test, which is M-chip. And literally it's the size of the credit card. You could hold it in the palm of your hand. It's a first ever commercialized microfluidic device for infectious diseases. And it has many, many fine channels. It would work great with blood, but there's going to be problems if you have a complex specimen like sputum to get it through those channels. But also there's a challenge of getting enough specimens with enough target into the system. So there has to be a concentration step up front as well, and it's read with the machine. So this is still, this has undergone evaluation in Africa, but it still needs further evaluations. And they have an HIV and syphilis combo test, and it's going to be less than $1. Now, with all these new technologies, especially if they're read with the reader, then there's the opportunity to put into the reader the connectivity to a central database, so that you could, even though you decentralize testing to huge areas around the world, you could actually have them beam up the results into a cloud or into a central database if it's all compatible with each other, and you could capture epidemiologic information and use it for surveillance very effectively. And also with these readers, you could actually put in a GPS so that you could pinpoint the location where the data source comes from. And so rapid tests or point-of-care tests allow, better linkage to care, because the results are immediate, they're right in front of the healthcare worker and the patient. And so that's been shown in Mozambique, in a lot of countries, that point-of-care devices actually make a lot of difference to linkage to care for HIV-infected individuals. Because I think in a study that was conducted in Mozambique and in Kenya, they found that 40% of the patients who tested positive for HIV are lost to follow-up. They never come back for their CD4 because they are scheduled for, months later, and they just forget about their appointment or they don't bother to come back. But having a CD4 test available at everywhere where you test for HIV would really make a difference about linkage to care. And then also, the whole electronic system of capturing data that is still to be exploited. And I think that it is going to be a really different way that we think about using technologies to diagnose and to